Good evening. I did not start this whole thing because I'm a huge Pippi Langstrumpf fan. It's because she says a sentence in the song, I mean, I know it was in German, hence we are going to get the English translation of it, that is very symptomatic for synthetic biology. I make the world around me so that it pleases me. I would like to welcome you to the lecture in Future Technologies about the topic of synthetic biology. We are going to deal with the origins of synthetic biology and the consequences it has. Oh, yeah, we turn it off. So, we're going to talk about the origins of synthetic biology, its relevance for science, and also its consequences for science and society within the next few years. And the sentence, I make the world around me so that it pleases me, very much points out what synthetic biology is doing. We are generating something, however we want it, that normally is up to nature. But we have to quickly start by looking at what we define as nature nowadays. Nature today is not what nature used to be about 50, 60 years ago. You see here on the picture the listener. The listener is not reality. The listener is an artistic piece by an artist from Australia, Patricia Piccinini, who is very much interested in bringing synthetic biology to the general population. So you might say, okay, whatever artists do, I'm not that interested in it. What are the facts? The facts are that there are companies already in this world where you can order a baby that has blonde hair, blue eyes, is good at sports, and is about 1.80 meters tall, and is just a great guy. Now, whenever you hear this, most likely you're not thinking about the technology behind it. Most likely you're going to be thinking, do I want this baby? And if I order the baby and it's not the baby I want, what do I do with it? If you order a car in the color yellow and you get a black car, you're not going to take that car because you wanted to have a yellow car. If you're going to order a baby that's good at sports, but all of a sudden it sucks, it can't even catch a ball, what are you going to do with it? Discard it? It's a little bit difficult if you talk about life. So those are things that we're going to discuss over the course of today, although the social relevance is going to be discussed a little bit more next week on Thursday. Today we're going to look at the origins of synthetic biology and mainly the way we use it in science nowadays to address important medical questions. Now, if we talk about biology, we talk about life, we talk about nature. I'm pretty sure that everybody in this room will agree with me that the picture you see there is nature. Mount Everest in the Himalayan mountain range, there's not too much man-made in this overall structure. Okay, if you look at the mountain more closely and you see all the garbage that the expeditions leave there and stuff, then it's already going to be a little bit of a different issue. So I guess this is nature. But what about this? This is nature. I mean, it's growing out there, the grass. But obviously, it wasn't like this 5,000 years ago. We did something to it, and this came out of it. Okay, it's beautiful if you like playing golf, but in the end, it's not really what it used to be. So is this nature with a trademark sign? As soon as we have nature with a trademark sign, of course, questions arise about patents, about who owns the rights, and all these kind of things, which is another issue that we're going to be discussing today. Now, this most likely is not nature. Okay, it's a live cat. But this cat glows in the dark. When you shine a UV light on it, it glows green. It's not born like this without interference of mankind. So this is what synthetic biology is about. So it's a little more than just the technology. It's something that we're going to have to discuss also in its social aspects. Now, one doesn't get around to defining the term at one point. And synthetic biology makes it possible for the biologists to become a creator. Now, this is a dangerous issue worldwide, because as soon as the humans start to create things that are alive, we have to ask the question, in what kind of moral, ethical background are they doing this? And it's a difference whether you're living in a country that's mostly Roman Catholic, or whether you live in a country that's Buddhist, or whether you live in a country where Islam is the major religion. They have completely different views on this issue. How far are we allowed to go? In the United States, you can order the baby. You can't do this in Austria. Yet. That's the big question. In order to get into the topic, I'm going to have to go back a little into the history of science that enables us to do the things that we're doing nowadays. I'm not doing this because I want you to learn the names of the people and exactly what they did. I kind of want you guys to get a feeling for the timeline that went by in research until the time when we really could start to manipulate life. And so what we're doing quickly in the next couple of overheads is we're looking at the research on genetic material, starting in 1869. Initially, this research was divided in two major groups. We had scientists that studied the function of materials that were believed to be genetic, and we had researchers, mostly chemists, that were purely interested in the chemical structure and the chemical composition of these kind of compounds. So the first thing, and back then the science was simply called biology and biochemistry. So the first thing we're going to do is we're going to look a little bit at the structural chemistry of these molecules, and then we're going to slowly bring in the functionality of these molecules. And then there was one scientific discovery, and this is clearly nowadays accepted by everyone. One scientific discovery that combined structure and function and enabled everything that we are doing today in biology. So the whole thing started in about 1869 when a German doctor who was very interested in infections, bacterial infections, and looked at inflammation of wounds, he found a substance that is present in every single cell that he looked at. And he just named this substance nuclein, because the nucleus in the cell, we're going to get to that a little bit later, is a special compartment within a cell where he found this. He had no idea what this was chemically. So people started to look a little bit closer as to what it is. Albrecht Kossel then found out that they are bases that are all containing a nitrogen, and they are ring-like structures. And the next guy then figured out, Theodor Levine, that it's not just the bases, but these bases are always attached to a sugar and a phosphate molecule. And from nucleobases, we came to nucleotides. And by now, we know those are the basic building blocks of our genetic material. Then came William Asbury, who was a little bit more interested, well, how do these molecules really look in a three-dimensional structure? So far, the chemists only could figure out what atoms, what parts of substances were present within this substance that they called nuclein. And he took this substance and crystallized it and examined it via X-ray crystallography. Basically, what you do is you have a big X-ray tube. It emits X-rays, and you send these X-rays through a couple of lead plates that have small pinholes in it, so you get a very directed beam of X-rays. And then you have the crystal of your substance that you're looking at, and behind the crystal, you have a photographic plate that collects the diffracted X-rays, and you get a blackening where there are X-rays, and it stays more or less clear where there are no X-rays that are hitting this photographic plate. And this is what he came up with, pictures like this. Now, we don't have to go into what this exactly means, but the obvious thing is this must be a highly symmetrical molecule. There are about 8 axes of symmetry if one looks at this picture closely. There's the obvious ones who are going vertically. There are horizontal ones. There's also some diagonal ones. So there's a lot of symmetry in this molecule. This was very interesting because normally chemical substances don't give X-ray diffraction patterns like this. It looks pretty chaotic on the X-ray film. So this started, like I said, Miescher in 1869, and now we are in 1937. Over 70 years it took us to get to this point. I mean, nowadays, this would take a couple of hours. If we look at the function, so we've discussed a little bit about the structure. Now let's look at the function. They had no idea. They just looked at substances that are present in cells and were wondering what they were doing. The first one that, although he didn't know it, but made an extremely accurate prediction of what this substance nuclein does, was Nikolai Koltsov, who said, this must be the genetic material, and it's a huge molecule that has two mirrored strands that replicate semi-conservatively, whereby each strand serves as a template for the synthesis of a new strand. This was in 1927. We don't have to worry about the term semi-conservatively and all that right now. The amazing thing is, he made a prediction that 30 years later was proven down to the point. This was simply by observation of things that occurred in nature. He didn't have any fancy instruments with which he did his science. But then, at the term between the First and the Second World War, medical research on bacteria became very prominent because so many soldiers died during the First War due to bacterial infections of the wounds that they got inflicted with during the war. And especially the British became very big in bacterial research, and Frederick Griffith worked with Streptococcus pneumonia, which is a bacteria that causes the infection of the lung, pneumonia, which at the time was a very, very lethal disease. About 85% of the people that got infected with Streptococcus pneumonia actually died of a lung infection. And he figured out, if one looks at Streptococcus as they are growing in the laboratory under defined conditions, they can look completely different. Each one of these little dots here is one bacteria, a Streptococcus. But under certain conditions, they can also look like this. This is also just one bacteria. Now, what he did is he made experiments to figure out whether he could change these, the small ones, into the big ones, or the other way around, whether he could change the big ones into the small ones. He was successful at doing this. He said, okay, there must be an information storage within a living organism that tells the organism what it looks like. And I can pass this information storage from one individual in my population to the next individual in my population. That's like if I'm getting a hard drive from you where there's some data on it, I put it into my computer and I can read out the data that's on the hard drive. So this biological hard drive somehow must be passed on from one cell to the next, or from one organism to the next. And then came the big time of the United States microbiologists that were funded with drastic amounts of money during the Second World War. Avery, McLeod, and McCarthy could figure out what inside the cell is this substance that is being transferred from one organism to the next and makes the recipient organism, look different. We know it a little bit different. It's heredity. You look the way you do because your mom and your dad looked the way they did. And while you were conceived, sperm and egg meet each other, your own individual DNA was created, and that's the hard drive from which you then evolved or developed. And they could clearly show that this information is being all stored in one single biological molecule, the DNA, the deoxyribonucleic acid. So with that, we knew that the nuclein that Miescher discovered almost 80 years before is actually the information or the molecule that carries information in the biological system. Basically, Hershey and Chase then were the ones that really fundamentally proved the hypothesis or the theory that was posted by Avery, McLeod, and McCarthy. And we're at a point now, the late 40s, the early 50s, where there was a lot of research that had been done about the structure of these molecules, a lot of suggestions and also conclusive evidence that this DNA molecule is actually the molecule that carries information in the biological system. But we had no idea how this worked. It was simply phenomenology. We knew this mechanism existed, But how it worked, we had no idea. And then came the one discovery that combined these two. And that was the discovery of the three-dimensional structure of the DNA molecule by Watson and Crick. In 1953, James Watson and Francis Crick published a paper. And I'm just showing this, not that we're going to read this, but first of all, this is an X-ray diffraction picture similar to the one from Asbury that we have seen. So basically, they didn't do anything else than what Asbury had done about 27 years earlier. And this is a Nobel Prize. They wrote one page for a Nobel Prize. I think that's efficiency. Most people nowadays are writing papers that are 15, 20 pages long, and there is absolutely no content to it. They did this a little bit different. They didn't do a single experiment themselves. They were just good observers. They traveled around the world, went to meetings where people presented their data with which they wanted to decipher the structure of the DNA molecule. Obviously, the other ones weren't smart enough to figure it out. Those two were. And that's them. James Watson is still alive and an active scientist today. Together with Maurice Wilkins, they got awarded with the Nobel Prize then 9 years later in 1962. Hence, sadly, Rosalind Franklin, who died 4 years before the Nobel Prize was awarded, she was not considered in this whole system. Okay, and this is what it looks like. I mean, we're not going to go too much into the chemistry. We're not going to go into the chemistry at all, because I'm not trying to make chemists out of you, nor do I try to make synthetic biologists out of you. But just to see, I mean, this is chemically as simple a structure as one can imagine. It looks like a rope ladder, basically, that's twisted around. And we have here these so-called base pairs. This is a term that we're going to need a little bit later on. So there are these nucleotides that were discovered by Phoebes-Levine. And basically what this molecule is, it's an alphabet. It's an alphabet that consists of 4 letters. We have the letter A, the letter C, the letter G, and the letter T. It's just merely an abbreviation of the names for these nucleotides that are there. And they are always lying opposite of each other. An A is always opposite of a T, and a C is always opposite of a G. And they form so-called base pairs. And we have 2 strings that are just running along in a seemingly random order of A, G, G, C, T, A, and so on. And on the other strand, we have the reverse of that, where if we have an A, we have a T on the other side. That's all it is. Tremendously simple. If we look at it, so with that then, actually, we have concluded this historical back view on DNA research. There was another experiment then by Messelson and Stahl that basically confirmed what Watson and Crick had shown, which I'm not going to go into any earlier. So now we knew how this whole heredity works in biology, but we still had no chance to use this as a technology because we didn't have any tools. We could look at it in biology. We could tell, okay, if there is a family where there's a lot of breast cancer, most likely the kids are going to have breast cancer also, especially if they are women. Those are things we knew, we could conclude from all the data that Watson and Crick had gathered, but we had no idea how we could interfere with the mechanism, how we could use this whole biological system for us, for a certain purpose in industry, for example. So we needed some tools. Now, in the early 70s, then, the first tools were discovered with which we could manipulate our genetic information so that we can produce something that helps us. The first thing is basically scissors for DNA molecules. They are called restriction enzymes. What we have here is the graphic representation of a small DNA molecule. And now there are specific proteins, enzymes, that cut the DNA molecule whenever there is a certain order of letters of nucleotides within the DNA molecule. So they have funny names, but in general, molecular biologists are not the most creative people on Earth, so they use bad abbreviations for molecules. So we have this DNA molecule, we cut it with this protein, it's called EcoR1, and then we get 2 DNA molecules. Now that's great, we can cut things apart. Doesn't help very much. That's a little bit of a destructive behavior that my 4-year-old son also has. He takes scissors and cuts everything to pieces that he finds in our house. It would be nice if he would have a tool where he could put things back together. So we need a glue. We also found a glue. DNA ligase is, again, an enzyme, a protein, that can stick DNA molecules that have ends that are compatible. Somehow they must look the same. We can stick them together. So this is awesome now. We can take DNA out of anything, let's say a plant. We can cut it to pieces, pull out the pieces that we need, and put it into another DNA molecule, let's say of a dog, and then glue this whole thing back together. It's not quite that easy because we need something to transport the DNA molecule. We need a shuttle vector. Naked DNA doesn't really work very well in a laboratory. So we needed something where we can put this in. So then the plasmids were discovered. This is all in the late 70s within like 2 years. Basically a plasmid, here we see a bacteria. Bacteria have a chromosome just like we do. We have more than one, but they have one. And they have additional hereditary information in the form of DNA, in the form of plasmids. Now plasmids, I can give you an example where nowadays plasmids are very, very important. I guess most of you have heard of the bacteria that become resistant to antibiotics. The classical problem nowadays in hospitals. You go into a hospital, have surgery. During the surgery, you get an infection with an antibiotic-resistant bacteria, and probably your initial problem was solved, but you have a new problem that's a whole lot worse, that you don't ever want to have. This information, why they become resistant to the antibiotics, is always present on such plasmids. So they are extra pieces of DNA that are present in the bacteria, and they contain specific pieces of information. We're going to look at the word genes a little bit later, that are not necessarily required for life-sustaining functions. So basically, they don't necessarily need it to survive. But if they have it, they can make life much more plush and nice. And they can be passed on from one bacteria to the next. So now we have a great system. We can take one of these shuttle vectors, one of these shuttle molecules, a plasmid, and cut it open with our scissors, restriction enzymes. We have a DNA molecule where we want to take a specific piece of DNA out. We can also cut this with our scissors. Take the right piece of DNA out of this whole mess of DNA that we generated by cutting it to pieces, and put it into the plasmid, and generate the completely, with the DNA ligase, we have a glue, and we generate a completely new piece of DNA that we can pass into another organism. Why is this awesome? Well, this is awesome because we can make billions of euros with this system. This is what pharmaceutical industries are living off nowadays. Because with that, at the end of the 70s, molecular biology, gene technology became a business model. And it was at this time when we had to find a name for this whole new science that all of a sudden came about. There was a big conference in Alamo, California, where the leading scientists that were working on DNA and molecular biology all gathered. And the sole purpose of this conference was, let's sit down and we try to figure out, what do we call this, what we're doing here? The first suggestion was, let's call it synthetic biology. They said no. They said no because at the time in the 70s, this synthetic biology, we're creating life, didn't go very well with the general public. There was not a politician in the world who could convince his parliament and his ministers to provide millions and millions of euros or dollars for a research area that creates life. So let's forget about synthetic biology. We call it gene technology. Okay, no one understands the term. It has something to do with technology. That's always nice. And that's it. However, the basis was laid for a big business model. These are the three scientists up here, Stanley Cohen, Paul Berg, and Herbert Boyer, that discovered these tools that we just talked about. Now, in general, you can have one assumption that almost 100% holds true. Scientists are extremely bad business people. So they needed another one that kind of knows how business works. That was Rob Swanson. So they realized that what they had is the possibility to revolutionize the medical market and the pharmaceutical industry. And this is actually quite interesting because this is a page out of Herb Boyer's laboratory notebook, where he describes what I just shown you earlier with the scissors, with the glue, and with the plasmids. And there is a sentence down here at the bottom that says, general applicability of the method. So what they did under the guidance of Rob Swanson, they submitted a patent to the United States Patent Office where they had patented the complete gene technology method catalog that existed of cutting DNA, gluing it back together, putting it into a shuttle molecule, and transferring it from one organism to the next. This patent was granted in 1978. This is the most successful patent in life sciences that ever existed. The patent ran until 1997. The profiters of the patent were Stanford University and the University of California at San Francisco. Over 480 industrial licenses were sold on this one patent. Over 4,500 mentions of the patent in scientific publications were made, and these two universities earned $255 million on this one patent. This is why Stanford and UCSF are the big medical research universities in the United States. They funded it with this one patent. Okay, so we have a business model. What is the first product from synthetic biology that became interesting for society or for the medical industry? It's insulin. Insulin, the medication that people need that have diabetes. Classically, insulin, and I don't know if everybody knows this, but it's not very pretty how insulin is being produced classically. Basically, it's isolated out of the pancreas of pigs. So you slaughter a lot of pigs every day. All you need from this huge pig is the small, tiny pancreas that's about this size. And from this organ, you extract the insulin. The bad part about this is it's very expensive. So we look for a method that's much easier, where we don't need to slaughter millions of pigs, and that is much, much cheaper. So what do? We do exactly what we showed with the tools of molecular biology. We take a cell out of a human, from a human pancreas, and of course this cell has the genetic information, DNA, in it. We cut this with restriction enzymes and pull out the information of the whole genomic DNA that's responsible for producing insulin in a human. I again, on the other side, have a bacterium that has this plasmid DNA in it. That's a nice shuttle molecule. Take this out, cut it also open with a restriction enzyme with our scissors. And then I just combine the two, the plasmid and the insulin here, and I produced a molecule that didn't exist like this before in nature. It's a new DNA molecule. I bring this DNA molecule now into bacteria. Normal bacteria, they're called Escherichia coli. You have trillions of them in your own body, in your gut, because they help you digest the food you just ate at the buffet outside. And without that, believe me, eating doesn't become fun. So then we have bacteria, and we just grow them in large-scale reactors. I mean, large-scale, I know there's some people from hardcore industries here. Large-scale for a biologist is like 10 cubic meters. I mean, so it's still big, but I know there's some industries, 10 cubic meters is like they don't even start to think about size. But while these bacteria are growing, and they are bacteria, but they have the human information for insulin in them, they produce human insulin. And this insulin then can be extracted from the whole bacterial culture, and then you package it and you sell it as human insulin. Nowadays, worldwide, the market for insulin is about 35 billion euros a year. So just from one medication, the pharmaceutical industry makes 35 billion euros a year. The overall revenue of the pharmaceutical industry, by the way, in 2013 was about 987 billion euros. So this is an industry that is not necessarily working on a small scale when it comes to money. Now, what can happen, though, and this is now where we're already seeing a little bit of the societal aspects of this technology. This system, as it's being depicted here, was performed by scientists in the United States, and the pharmaceutical company Eli Lilly bought the rights to produce insulin this way. However, they didn't really believe in the technology, and they sold the rights to another pharmaceutical company, Genentech. Genentech today, by the way, is a partner of Roche Pharmaceuticals, and they are one of the 5 big players in the pharmaceutical industry nowadays. And they were looking worldwide for production sites where they could produce human insulin just like this. One of the countries they were thinking about naturally was Germany. Not because Germany is such a great country, but classically, the Höchst industry in Germany was the largest insulin producer up to the 1970s, 1980s. So they figured, okay, there is already a lot of know-how about the insulin production. They wanted to do this near Frankfurt, which is in the state of Hessen. At the time, so they put a proposal to the government of the state of Hessen in Germany whether they could produce, build up a production plant to produce human insulin with these genetically modified Escherichia coli. At the time, the Minister of Environment in Hessen was Joschka Fischer. Joschka Fischer, the former Minister of International Communications and whatever else from the Green Party, got this proposal, didn't want to make a decision, and put it up to the parliament whether this production plant would be constructed. The parliament said, we don't know, let's ask the people. The people, with an 82% no vote, rejected to build this plant. Why? Because they were afraid that a genetically modified organism is being grown in their neighborhood in extremely large amounts. And no one knew about the consequences if one of those reactors broke, if those bacteria get out into, now we're back to the nature term. And all these resentments led to the fact that in Germany, no insulin was produced anymore with the new technology. This was a huge blast against the pharmaceutical industry in Germany. They took about 20 years to recover from this one political decision that was made by the society, by the people of Germany. Ever since the late 90s, 1998, since Sanofi bought Höchst, they are back to be the biggest insulin producer in the world, and nowadays they produce it like this also. But a decision on a technology can change the course of the economy in this country dramatically, as we can see from this example. Okay, so much for the whole overall picture, what this synthetic biology coupled with the pharmaceutical industry is all about. What I want to go into next is I'm going to give a little bit of basic scientific background so you get a little bit of a better feeling about the makeup of an organism and what this DNA molecule is really doing and how it works. Because in the end, then today, I'm giving you 2 examples of synthetic biology as it's being used in research today. So actually, this whole synthetic biology thing is not very complicated. Because no matter which of the organisms that you see coming up here, the basic function, no matter if it's a human, if it's a dog or a cat or anything else, the basic function and the basic structure is exactly the same. And I just want to go back with you now from the finished organism down to the level of what really happens at the genetic level, down where this DNA molecule that stores all this information is happening. So this is what we look like, by the way. We have a skeleton that provides our structural support. Otherwise, we would just basically fall down and would be an ugly blob on the ground. It also allows us to walk around and to point out things that are great or not great. And we have the organs. The organs provide, of course, the very important life-sustaining functions, and they enable us to do a lot of things. Now, if we go down another level and look at the organ, an organ is made of different tissues. So within each organ, there are several different tissues, and these tissues all perform somehow a specific function for the organ. Now, the tissue itself would also be just an amorphous blob, so we need something that structures the tissue. It's a protein scaffold that's being produced by the tissue itself. So you have to imagine this as a growing structure. We start somehow with one of these ugly little Pac-Man structures with one of these gray eggs in it, which is a cell. And it produces basically proteins that it sends to the outside, and you build this large scaffold so that tissues are not just amorphous blobs, but that they can have structure. Now, if we look at the tissue in a little bit more detail, we see that the tissue is made up of several cells. Now, all these cells in one tissue are exactly alike. Yeah. So all their neighbors are like identical twins. This is then the cell. So if we look at the cell a little bit more closely, we're down to the basic building block of an organism like a human. This is what you're made of. A lot of them make up what you are. A human has about 3.7 times 10 to the 13th cells. Almost every single one of these cells, by the way, contains your genetic information. So it contains the whole information storage that is required for life. Huh? And this information storage compartment is this part in here, which we call nucleus. It's a word that comes from the Greeks. And inside this nucleus, inside of almost every cell in your body, we have the DNA molecule, so our basic information storage, our plan of what life should look like in you. And this DNA molecule is actually quite interesting because I told you it's like a linear molecule that looks like a rope ladder. If you pull the DNA from one cell out and you stretch it out, it's about 2 meters long. So you have 3.7 times 10 to the 13th cells that all have DNA in it that's about 2 meters long. That's a lot of DNA, but you still consist of a lot of other stuff. But DNA is a major part of what you are. An analogy is, for example, and it must be packed into this small nucleus that's only about 0.002 millimeters in diameter. That's like if you want to pack 40 kilometers of string into a tennis ball. You will pack a long time until you have these 40 kilometers inside a tennis ball. I've tried it with 1 kilometer and I didn't succeed. So this takes a while. Now, just quickly again, what this DNA molecule looks like. All the information of an organism, the way we look, whether we are susceptible to certain diseases, meaning do we get prostate cancer at age 50, yes or no? All these things are basically stored in our DNA. Yeah. Overall, and I told you about these base pairs. So one of these G letters and the C letters, they always form the pair. And we have 3.2 billion of these base pairs in each one of our cells. So this 2 meter long string is basically a succession of 3.2 billion letters. That is just quite long. And it is stored in the nucleus, and we already heard about Watson and Crick that got awarded for the Nobel Prize. So, now how does this really work in our system, in a biological system? Here we have the DNA molecule, just represented now as a bar. And I already told you earlier this word gene. Now, what is a gene? A gene is a functional unit of our information storage, of the DNA. And one gene, and so our genes are just spaced somewhere randomly across this whole genetic information molecule. One gene encodes for one protein that has a specific function. Overall, you need about 25,000 genes, so you look and function the way you're sitting here right now. That's all contained within our DNA. Now, how does it work? It works similar to when an architect builds a house. We have our DNA where the genes are on it. Like I said, the genes are the functional units. From those, our proteins are produced. Just like an architect starts to draw a plan of a house, and he has different rooms, and when construction begins at the house, you start, of course, first at the basement, and you go up, and eventually you build a roof on top of this thing. And in the end, you have a functional, hopefully a functional, but you have a finished house. Whether the roof leaks or something, that depends on who you use to build it. In biology, it's the same thing. So we have the plan, we have the individual building blocks, and in the end, you have the finished organism. And that hopefully works quite well. So this is basically all that we need now in order to really start getting into the science of this synthetic biology part. Okay, now let's talk about two projects that made quite a stir in the last 5 to 6 years in terms of synthetic biology. Like I said, today I'm going to concentrate on how the synthetic biology is used for science. Next time we're going to very much concentrate on what does this mean for society, what are issues that we're going to have to discuss in society in order to deal with this technology in the future. Synthetic genomes. Now, the completeness of the DNA that you have in one of your cells is called the genome. Yeah? So your genome is the plan as to what you look like and all those kind of things. Now, the big question is, of course, your genome was made by biology. It was made by your parents, their genomes was made by your grandparents, and it goes back and back ever further. Can we do this completely synthetically in a lab? So can we make a genome man-made? And if we make it, does it function in biology? That was a big question. And the question was answered then in a huge research effort and was published in Science in the July of 2010. So when you're starting to build or synthesize, make mechanically a human genome, you're not going to start with a human genome because it's a bit big. Take an organism that has a smaller genome because that's easier to test. Plus, I don't know how the acceptance would be if I'm going to sit in the lab upstairs and say, no, I'm making a human genome, and we're going to have a little freak running around after a couple of months. So they started with the smallest biological organism that can actually autonomously replicate itself. So basically, this is the smallest organism on Earth that can live all on its own. Almost all on its own. It's called a mycoplasma. These are single cells. So basically here we see 7 mycoplasma. 1, 2, 3, 4, 5, 6, 7. And each one of these is its own organism. And it only has a genome. I mean, there are different types of mycoplasma. But if we look at it from 580,000 base pairs to 1.3 million base pairs. So compared with our 3.2 billion base pairs, this is minimal. Very small. So what they did is the following. They had machines create, and by the way, what they tried to rebuild is the genome of a mycoplasma that has a genome size of about 1 million base pairs. That was the goal that they set out. So what they used is they used machines that can synthesize small pieces of DNA. Now, we don't have a machine, even nowadays, 4 years later, where you can just say, synthesize me a 100,000 base pair long DNA molecule. This is not possible. We can't do this. This has a lot of technological and chemical reasons. So they produced small synthetic DNA fragments that are about 100 base pairs in length. Now, if you have 1 million base pairs in the whole genome, and you synthesize pieces that are 100 base pairs in length, you need 10,000 of those in order to do this. So they synthesized 10,000 different DNA pieces with exactly the same order of letters as it occurs in nature. So now you have 10,000 pieces that you somehow have to put together. You can't do it in one step. What they first did is they always put 10 of these pieces into a bacteria and let the bacteria do the work. This is actually the great thing. They didn't have to do too much work for that. They just took 10 pieces that are usually in a genome right next to each other, and you put those 10 pieces of DNA just into a bacterium. And the bacterium automatically, magically, puts the 10 pieces in the right order and combines them. So then you generated from 10 108 base pair long fragments, 1,080 base pair long fragments. So then you only have 1,000 of them. You already reduced the number from 10,000 to 1,000. Now again, you have to put these 1,000 different DNA pieces that are all 1,000 base pairs in length together. Again, you do the same thing. You just put 10 of these that are right next to each other into a bacterium, and the bacterium just grabs those and puts them together in the right order. So nature, in a way, is a great thing because it takes away a lot of tedious work from you. And then you get pieces that are 10,080 base pairs long. 109 of them. The reason why the math isn't always completely correct is because they have to overlap a little bit and you lose base pairs and things like that. But that's, I mean, not necessarily an important fact for this whole story. So now you have 109 base pairs, 109 pieces that are all about 10,000 base pairs in length. What do you do next? Well, they thought, hey, we put those in bacteria again and it's going to work. Well, they did, and it didn't work. Because for some reason, these 10,080 base pair long pieces, 10 of them, that's too much for the bacteria to handle. All of a sudden, they can't put them back together. So they had to find a new way. Well, they used another organism that's a little bit bigger than the bacteria and tried whether they could put these pieces together. And they found one. They used yeast. The classical yeast that one uses to bake bread or that one uses to brew beer. They put these 10 fragments that are 10,080 base pairs long into a yeast. All of a sudden, the yeast could do it. They grabbed these fragments, put them together in the right order. So they got 11 fragments that were each 100 base pairs in length. And then came the tough part. This whole project took about 7 years to do. The point where we are right now, that was done in about 2 years, a little less than 2 years. The next step took 5 years. Because now you have these 11 base pairs, these 11 fragments that are each 100 base pairs in length, and you have to put those together. Again, they put it in yeast. Nothing happened. Well, what do? Try a bigger cell. Oh, let's use a human cell. Put it in there. The pieces got destroyed. That's the classical nature of human, destroyed. So they had to somehow do something else. They didn't find anything naturally occurring in nature that would put these stupid 11 pieces together in the right order. So they took yeast, and basically what they did is they genetically modified the yeast. What they did is they put a lot of new information into the yeast, took other information out of the yeast, and basically they just tore this yeast apart, put the new yeast together, and eventually they got a yeast where they could put all 11 pieces in, and all these 100,000 base pair long pieces were put together into one genome that was then 1,077,947 base pairs in length. So they had their genome. It was synthetically made. Whether it works, they had no idea at this point. It's very interesting, though. So how do you know that the genome that they made is the synthetic one and not the one that you got from nature? Because obviously now the next step is they have to prove that their synthetic genome works in biology. Now, the easy thing would be just take the real biological one and put it in a cell and say, hey, it worked. So you cheated a little bit. Well, no, they didn't, because what you have here, there's 4 regions that are called on here WM4, WM2, WM1, and WM3. They put information into those 4 spots that clearly identified that it was their synthetic DNA. What they did, for example, in WM2, they put the names of all the researchers that were part of the project in DNA code into this genome. It's geeky, I know, but they did. WM1 is the first passage of the book of Genesis that they included here. And then there is some additional information that they included in here. So this synthetic genome can clearly be identified later on as the real synthetic genome. And so this is the way they did it now. Here is their yeast cell with their artificial genome. What they did is they pulled this genome out of the yeast cell and had it free, open, without a cell around it. Now, the genome it came from a mycoplasma called Mycoplasma capri. So this is basically just the name of this mycoplasma. So we're just concentrating on the name capri. At the same time, they took another mycoplasma that's called capricolum and extracted the natural genome from this guy, from capricolum, and had then a naked cell without DNA. And now they combined their artificial genome from Capri and stuck it into the empty naked cell of capricolum. And now is the question, what is the result? Does this organism that's being created live as a Capri or as a Capriculum. And the result was it actually lived as a Capri. So obviously this worked. The genome that they completely synthetically made in a laboratory is a fully functional genome that can sustain life functions. This organism now lives for many, many thousands of generations, and it's still running, so there doesn't seem to be any long-term effect why it might not work. This was a very important proof of principle. Now, how scary does this get? When are we able to synthesize a human genome with 3.2 billion base pairs? And we can basically put all kinds of information in there, and we're going to have exactly the person that we want. Well, it's still far away. The next step that the scientists are working right now, they want to make the synthetic genome of the bacterium Escherichia coli that we already talked about, the little guy in the gut that helps digest us. It has a genome of 4.2 million base pairs, so about 4 times the size. So far, they haven't been able to do this. So it's not like we're going to have to be worried that humans are going to be replaced by synthetic genomes in the near future. However, the technology is going to advance. And I just heard at a meeting a couple of weeks ago in Vienna that they are close to the synthetic E. coli genome now. It seems to be that it actually is working, but it's not published yet. Why are we doing this? I mean, a lot of money is going into this research. 40 people worked on this project for about 7 years. Why are we doing this? We are interested, science for a long time, for the search for the minimal genome. Now, what is the minimal genome? We, as we sit here, with our 3.2 times 10 to the 9th base pairs in our genome, we have a lot of DNA that we probably don't necessarily need to just survive. Now, the minimal genome is that genome that just barely contains enough information so that an organism can survive. So you take everything away that you don't need, and you still have enough genome present to that this organism can still survive. It's like if you read a book with 1,000 pages and 980 of it are complete trash. So you read the 20 pages and that's the essence of it. That's what they're trying to do on a DNA level with genomes. There are 2 approaches that one can use. This is basically a genome where all these little boxes here are genes that are present on this genome. And so you just delete a few of them, where you think they might not be needed for life, and you check whether an organism with this smaller genome can still survive. That is one approach that you can use. The other approach is the following, that you basically just take individual genes that you isolate from different organisms, put together a genome, and check whether that organism can survive. Now, the interesting part of the whole slide here then is, what are these? These individual bricks, these genes that one can actually put into a genome. These are biobricks. Now, biobricks are reality already. A biobrick is a building brick of synthetic biology. Basically, it's a DNA molecule that can perform a clearly defined function. For example, there is a biobrick that produces a protein that can digest palm oil. There's also a biobrick that can synthesize, or there are biobricks that can synthesize colors, very nice colors. There are biobricks that can synthesize insulin, the thing that we saw at the beginning or earlier with the whole insulin production in bacteria. That information of DNA that's being pulled out of a human cell and being put into a bacterial cell, that's a biobrick. And what one wants to do is actually do the same thing as, for example, in electrical technology. We just want to create a system where we have a big shelf with biobricks, and I pull bricks off of the shelf, combine them with the glue, and I generate an organism that can perform a function that does exactly what I want to do. There are databases for these biobricks that are openly accessible. So, for example, one at MIT, at the Massachusetts Institute of Technology, the biobrick database has about 165,000 biobricks in it. So you can just pull out biobricks from this database, You write them an email, what you want to do with it, and they send this biobrick to you, and you can plug it into your organism that you're working with. So it's like playing Lego. Yeah? You have a lot of Lego pieces, and you just start to build stuff together, and it's awesome, because a lot of times it actually works just the way you're thinking about it. This is the future of molecular biology or of synthetic biology, because we want to be able, just like with all other technology. I mean, what does the automotive industry do? They also just pull more or less things off the shelf and put them together and build a new car. Okay, you have to make adaptations and all those things, but overall, this is what synthetic biology is striving for. A system where you can just plug and play. Okay, the next project, and it's the last project that we're going to talk about. We're talking about chimerism. Now, chimerism is something that fascinated humans for many, many thousands of years. What is a chimeric? There are a lot of chimeric humans out there. And considering not all of the people are here, but if the normal 120 people would be sitting here, I guarantee you that statistically one chimeric would be sitting here. He or she doesn't know it, because most of the time it doesn't necessarily have such an obvious consequence as it does with these people. The other people that are sitting here that are not chimeric, I'm not a chimeric, I know, because I tested myself for it. Well, it's interesting to know whether you're a chimeric or not. I don't know what you're going to do with that information, but I was just wondering. All the DNA that we have from every single cell, we're going back to the 3.72 times 10 to the 13th cell. Almost all of these cells, like I said, have DNA in them. They have the human genome in it. And it's the same in every cell. Yeah? So you have one DNA that is the same in every one of your cells. I mean, otherwise, for example, DNA testing in criminal activities wouldn't work. Yeah? So this is one of the bases. Now, a chimeric has more than one type of DNA in it. It can be 2, 3, or 4. So this is, of course, a drastic case, because obviously this person is of probably African descent. The parents, one of the parents probably was not of African descent, and something during the early development of the human inside the mother happened to where he doesn't just have one type of DNA, but he has 2 types of DNA. Now, this person doesn't have any problems in their normal life functions. I mean, in this case, of course, it's clearly visible, but it's not like they die earlier or anything. They just live completely normally. But chimerism is something that has always fascinated us. For example, if we look in mythology, we have these creatures, the lion and the goat and all these kind of things. And a lot of times they have, the snake comes out as the tail and so on. This is the classical thing that for thousands of years have fascinated us. So it still fascinates us, because what can we do? This is a great title where I'm sure everybody knows what's happening. Forebrain engraftment by human glial progenitor cells enhances synaptic plasticity and learning in adult mice. I'm sure everything's clear now. What they did is they made a mouse with a human brain. That's what they did. Basically, it's just a little bit more complicated the way they set it up there. Now, 2 things, or I'll tell you why they did it in just a second. There have been many, many chimeric animals that have been generated in research. A classical chimeric animal that has been generated in research, for example, is a pig with a human pancreas. Okay, it's nice. We have a pig with, for example, human bone marrow. Why do we do things like that? For example, the pig with the human bone marrow. Everybody's heard of the pig flu, the bird flu, and whatever else. Now, obviously, the pig flu didn't come from humans, because then it would be the human flu. So it comes from pig. So how can the pig flu virus infect human cells? One didn't know how this works. So what they did is they made the pig with the human bone marrow. So basically, the whole blood system of this pig was from humans. And then they infected these pigs with pig virus. And then the mechanism was found out as to how these viruses can also infect human cells. So that's why one does things like that, just as a quick information. Now, there hasn't been a single ethical problem with a pig that has a human pancreas or a human bone marrow. It happened, it's great for medical research, we get a lot of knowledge, but this wasn't an ethical problem. These animals right here, they made a huge stir, especially in the United States, because these two researchers, Steve Goldman and Mike Nettergaard, they are married to each other. They're working at the University of at UCLA. When this was found out that they are planning an experiment to make a mouse with a human brain, every ethical commission, every church institution in the United States started a huge campaign against these animals. However, a pig with a human pancreas, no one cares. Now, again, we're getting to an organ now that is not just an organ like the heart that pumps blood or the pancreas that produces insulin and all those kind of things. We're talking about the brain. The brain gives us a little bit more than just life-sustaining functions. It forms our personality. It's a very, very personal morale issue when you do something like that. So, why did they even do it? Well, now we have to be a little bit very self-confident as humans. Obviously, when humans are being compared with any type of other animal that's living out there, we have a couple of advantages in life, or let's at least say we can perform functions that other animals can't. So somehow we have an evolutionary advantage compared to other animals. Now, but if you look at the structure, as I showed you earlier, it doesn't matter if it's a dog or a human, we look more or less the same. Our cells look more or less the same. You can't look at the microscope and say, this is a dog cell, this is a human cell. They look the same under the microscope. Almost. There is one cell that is clearly different in a human from all other animals. And it's a specific cell in the brain that's called an astrocyte. An astrocyte is a cell that basically has these nice little extensions, and they have little arms and knobs up here. And if we look at human astrocytes, they differ in two main points compared to all other animals that are out there. The extensions of the astrocytes in humans are much, much longer than from any animal out there. No matter even if you take a big animal like a horse, they're still smaller than the ones in the human. And our extensions in the humans are much fatter than the extensions in other animals. So we have astrocytes that are longer and fatter. Now, it's always been an idea that this is the reason why we have the abilities that we have. But it had never been shown. There hadn't been any experiments where one could do this. Just quickly, how does our brain basically develop? We have a stem cell, a neural stem cell, and you form two different precursor cells. So this is just basically a time-wise development of the different cells that we have in the brain. And you get a neural precursor cells that forms the actual neurons. Now the neurons are the ones where our nerve signals travel through. Yeah. And then you have the glial precursor cells. There you get oligodendrocytes and the astrocytes that are in question. What do these astrocytes have, or what kind of function do they normally perform? They support transmission of signals in the neurons. So you put your hand on a hot plate, you immediately pull it back because you're sensing, oh, it's hot. It's not just the neurons where the signal travels through, but the astrocytes are actually assisting the neurons in propagation of this signal. And they give structure to the brain. So they also have a structural function that's very important, and they regulate the blood flow in the brain by constricting the blood vessels. Basically, these long extensions can grab the blood vessels and tighten up, so no blood can flow through it anymore, or they can loosen up and blood can flow again. So that is the function of these cells in our brain. Now the question is, if we make a mouse where all the astrocytes in this mouse are human, is this mouse all of a sudden going to be the genius mouse, and it can read and it can talk and it can do a lot of other things? So this is the basic question that they asked. This is a picture of the astrocytes. The astrocytes are the red ones here. The green ones are the neurons where the signals travel along, and the red extensions are the extensions from the astrocytes. And this is a human astrocyte, so it has very, very long extensions. This, in turn, is the astrocyte from a dog, and it has much, much shorter extensions. And also, if one measures the mean diameter, it's a bit hard in this resolution. Our extensions are also fatter. So this is basically the background to this whole thing. So how do we make a mouse where a whole cell type in the brain is human and not mouse anymore? It's a little bit of a gross procedure. You take human embryo brains. Those are aborted embryos where something went wrong during development and they either were aborted by the mother naturally or where the embryo threatened the life of the mother and the embryo was taken out. So you take these brains and basically you put them in a mixer and mix them up. You get the cells then and you culture these cells. Basically, this is nothing than a round dish and it has a specific solution in it where the cells get food from and they can live on in this, we call it medium, so in this food. So now what you do is you let these cells go through a specific machine that only pulls out the human astrocytes. Of course, we took a whole brain here, so there's a lot of cells in there that are not astrocytes. But the machine, you can specifically pull out only the astrocytes. So we pull out only the astrocytes and put them back into one of those cell culture dishes with a medium that provides food, and they can continue to grow. Now we genetically modify these human astrocytes with the green fluorescence protein. Basically what we do with that is we mark those cells because they're always going to fluoresce green. So if I put these cells into a mouse, I know whether I have a human astrocyte or not because I have green fluorescence in the mouse brain. And then you xenograft them. Xenograft means you have these cells here, the human astrocytes that are all green fluorescing. In a needle and a syringe, you have a mouse into the brain, inject, pull out, you have a mouse that has human astrocytes in it. And it is really that brutal as it sounds. And then what you do is you make experiments with the live animals, or you basically kill these animals that you've injected with those human astrocytes. And first of all, look, do those human cells grow? Do they grow where normal astrocytes would grow? And you do a bunch of tests. And a couple of those we're going to look at now. So the first thing you're interested in is, of course, when you do something like this, injecting human astrocytes into a mouse brain, you want to know, can I find these cells in the brain? Are they actually even growing in the brain of the mouse? So for that, the mice have to be sacrificed. The brains are taken out. They're being cut into very thin slices, and they're being put under a microscope. Yeah. Yeah, I'm going to get to that in just a second, but thanks for the question. The first thing we see is, in these mouse brains, all of a sudden, we do have the human cells. And they are obviously astrocytes because they have their extensions. The extensions are, one can see it in this picture, but I'll have another one later. And they look very much human. Now, this mouse already had astrocytes. The very surprising thing is you can hardly find mouse astrocytes anymore afterwards. There must be an astrocyte war going on in there, and the mouse cells lose. So in the end, you almost only have human astrocytes in this brain. It was a very surprising finding also for the researchers that did the experiment, because they were expecting a mixture. But it's hardly a mixture. You find less than 5% of human astrocytes in there. So, okay, so we can find these cells. That's nice. So obviously, the basic idea works. The transplanted cells are growing in this mouse. The next thing is, are they also present in the brain where we expect them to find? I mean, it doesn't help us if these astrocytes are growing anywhere, but they're not growing where we expect the astrocytes to find. Again, they took the brain out, and they basically made sections through the brain, cut it into thin slices. Here we have the forebrain. This is the hindbrain, and then we just cut slices. This is basically what one of those slices looks like. And each one of these dots here represents an astrocyte in the developing brain. So this is still a quite young brain when the mouse is only about 3 to 4 weeks of age. And this right here is in the brain when the mouse is about 2 months old. These astrocytes, the humans one that we inject, are exactly located where we expect the astrocytes to find naturally. So this all looks quite good. The distribution of the astrocytes in the mouse brain is the same as the mouse astrocytes would be distributed in the mouse brain. So, are they still fatter, the extension? That's the next thing. So basically, they just measured the extension in these cells and looked at whether the extensions are, how wide or how fat the extensions are. Here we see the diameter of a natural, normally occurring mouse astrocyte. And here we see the diameter of the human astrocytes in the human brain, and this is the diameter of the human astrocytes in the mouse animal. They're exactly as fat as they were previously. They remain exactly the same size as they are naturally in the human, and they're overpowering in size the mouse astrocytes by almost a factor of 3-fold. So even that works. The size stays the same. Okay, good. Well, now we're going to have to think about, do they function better, those mice with human astrocytes in it? So the first thing you do is one looks at whether calcium secretion stays the same. Quickly as a background. If you get an impulse where you're moving, so basically you put your hand on a hot plate and you're pulling back, the neurons that relay the signal from your hand to the brain cause in the brain massive amounts of calcium to be released. You need this calcium in order for you to actually notice that there is a pain there. So this all, of course, works on a microsecond scale. Now, the trick is that humans have a much, much longer period of calcium secretion than a mouse does. So what they did is, do the human astrocytes in the mouse have the long calcium secretion period as the normal human astrocytes in the human brains do, or do they only have it for a short time as the mice do? And this is basically what they measured. They measured the speed with which calcium can spread from these astrocytes. You do this by isolating these cells out of a brain section, or you take a brain section, you lay this brain section under a microscope, and you direct a UV beam directly onto the cell. And this ultraviolet light beam induces the cell to release calcium. And then you measure the speed with which the calcium is traveling. And basically what you see, again, the mouse calcium travels at a speed of 5 micrometers per second. Yeah? In the normal human brain, the human astrocytes have approximately 13 to 14 micrometers per second. And the funny thing is, the astrocytes, the human astrocytes in the mouse brain, they're even performing better than the normal human astrocytes in the human brain. They are having a speed of about 16 micrometers per second. So in that aspect, they even seem to work better in the mouse brain than they normally do in the human brain. This is very much an indication also for effects of learning. Long calcium secretion means that we have a long-term electrical potential in the neuron, and that is related with learning capabilities. And so this is the last one that we're going to go over. Are they actually really better in learning? What you do here is, first thing, the good news is we don't have to kill the mouse for this test. The maybe not so good news is you don't want this done to yourself. What you do is they get a hippocampus-dependent learning test. So learning is very much dependent in humans, as in all animals, on the activity that is happening in a specific part of the brain called the hippocampus. So what they did is, these cells, neonatal graft, means the mice were injected with the human astrocytes, and then they were allowed to live for about 6 months until they're old enough to where the brain is completely developed, with the human astrocytes in it. What you do now is, you take this mouse, put it into a specific cage, and you let it sit there for 3 minutes. Then you play a sound for this mouse for 30 seconds. And as soon as the sound stops, you apply an electrical shock to the mouse. And the electrical shock is being applied through a metal plate that is basically the bottom of the cage. So basically this mouse is running around on a metal plate. And as soon as this sound stops, they get an electrical shock for 2 seconds. And then what you do is, you take the mouse out of the cage and let it sit for 3 minutes. And then you put it back into the same cage. Again, let it rest in the cage for 3 minutes. Nothing happens. The sound starts for 30 seconds. And again, they get an electrical shock. After this treatment, they're taken out, they're being put back into their cage, and you do repeat this for 4 days. The first thing that's very obvious is, what mice do is, already after the first electrical shock treatment, if they hear the sound again, just a couple of minutes later, they immediately freeze. It's the so-called freezing behavior that all animals have that are prey for other animals. What does the freezing behavior simulate? It simulates I'm dead. That's what prey animals do if they're under stress. Yeah, they play dead. Of course, they don't know that there isn't a big cat sitting next to the cage that wants to pull them out, but they see this as a threatening situation, so they freeze. And if one looks at the freezing behavior, which is an indication of how well they learn from the situation that they're in, and compares it to the normal mice, we see the following. That the normal mice have this is percent freezing. Basically, you film them, just like I'm being filmed now, and then you count the pixels where they're moving. The more pixels, basically when they move, of course, you have more pixels that the mouse is in. The less they move, the less pixels are. And that's how you get this percent freezing here. We see the normal mice, the unengrafted mice. So those are the natural mice, the way they occur. They basically have hardly any learning behavior. It's the same as on day 1 as it is in day 4. So nothing happens at all. If we look at the mice with the human astrocytes, they have a tremendously steep learning curve. After 1 day, their freezing behavior already doubled in time compared to when they received the treatment for the very first time. And this goes up to almost 3-fold the freezing behavior that they exhibited on the first day. So they do have a much better learning curve than the mice that didn't have the human astrocytes. The other thing that they can also do is, how do they deal with the completely new situation then? Basically, this treatment was being performed for 4 days that I just explained to you. On the 4th day after the last treatment, the mice are being pulled out of the cage. They're allowed to rest for 10 minutes, and then they're being put into another cage that looks exactly the same as the first cage, but it cannot apply an electrical shock. The mice with the human astrocytes are immediately able to recognize that this is the same dangerous situation that they were in before, and they immediately have the freezing behavior. The normal mice that do not have the human astrocytes, they're being put into this other cage and they're starting to run around like nothing ever happened to them. So the mice with the human astrocytes are also able to distinguish between different situations and can apply things that they learned in one situation into another situation. This is what's being met back here. This basically what they did here is a discrimination ratio. That's what this graph back here shows. So in the end, they made a mouse where one cell type in the brain is completely human. This mouse, so the cells grew, the cells remained the same as if they would have grown in a human mouse brain. All that works. And if they looked at the learning behavior, they could see a drastic increase in learning ability and also in the able to discriminate between different situations. They made a lot of other tests to basically show that this is really the way it was. That's synthetic biology, because this mouse with the human brain cells doesn't occur in nature. We are creating a system that we can use, in this case, for research purposes. Now, does this mean that it's really the reason why we're superior to other species, maybe because our astrocytes are different? This is a conclusion that I would not draw if I were the scientist, but they could clearly show that something in these mice is different from the naturally occurring mice. Okay. They do learn faster. Next Thursday, we're going to talk about basically the impact of this technology on society. So does it really have an impact? What are things that need to be discussed in society? Is it okay to have a company where you can order your baby? What does society do with this possibility to order babies and those kind of things? Now, are there any questions?